Uh, with my family. I think it's the most zero error defense. I don't find the error enough. I don't find the error in the algorithm.
Except the Wi-Fi subject. No, the area, but the whole area. Just this little.
Yeah, we're good. We're good on that.
Very good.
Some breathing and that can get you through.
You know this thing that we do. You have a calendar says we yes. If you don't know, then it's a defense. I'm sorry. Yes, I understand. Yeah. Oh, sorry. I think it's true. I hope Manon will find a way to. I sent her the YouTube link yesterday. The shame that we didn't find the. It's really important. And then I don't know. She looked at the time. Yes, I can imagine that. Yes, I know you can think it's happening. I think more than I just all the same world. Comme le lien, il est disponible là, sur YouTube. Non, c'est parce que Shaima hier, elle m'a demandé, je lui ai dit, je pense qu'il doit être quelque part. Non, je sais pas son mail, la chaîne.
On peut partager sur le groupe des PhD, mais je sais pas son numéro de chaîne. Je crois que sur mon YouTube, il n'y a pas un truc qui me dit que je suis en live et tout.
Imagine that. If I'm able to.
Justement, j'attendais la réponse. Le
Yes, it's your answer, my friend. It's on my YouTube. It's good.
Yeah, that's
You didn't check. You didn't check your screen. Yes.
Finally, we're here. Finally, it's bonjour. Bon, I don't know if you're talking with the book or the big president of the jury. I didn't know it was my way. I asked him before. Okay, so both, yeah, you can application wherever you want. Okay. There is a description of the members of the jury every picture. And I mean, everybody can sit over there just on the laptop and you can sit wherever you want in any, yes. Perfect. Okay, okay, yes.
Perfect.
You have to open the Zoom again. You're good?
So you're.
I'm sharing, yes. I'm sharing.
Juste à droite, à gauche.
That next one, I have to move first the. J'ai vu sur la. Bah, je suis à Sarah, comme là. Comme sur le. Ça va?
Ça a toujours fou de gens de revenir.
C'est pour les gens de. Donc, ça permet d'avoir les rangs du chemin, et puis alors, c'est une chose il y a des roulettes, faut pas le dire. Attends, je vais alors, vas-y, tu une prise des graisses, ensuite, ranger sur les de stress. Ensuite, nous allons appeler il y a Julie Joyce, c'est ah ouais, mais je pense que je vais m'adapter quand vous avez place. Ok, moi je peux vous. Ouais, voilà. On va demander aux ensuite, on a 10 minutes. Oui, on a 10 minutes. You got 10 minutes. Okay, so with you? Yes. We still have, I don't know if we have people coming from India, so maybe we have to wait. Yes, yes, sure. We're still waiting for Professor Josh to connect, so we are not open and we're gonna have the professors here soon. Huh?
Ce soir, nous allons faire le séance.
Okay, so we are waiting for jury members. Remote? No, Anastasia is here. Hi, Anastasia.
Yes. Hi, everyone. Can
Nice
you
to
hear
meet
me?
you. Yes. Yes.
Great. Nice to meet you, even online.
Yeah. And so we are also waiting for Julie Joss, who is in Montpellier. Okay. Minutes, I hope she already. We cannot start without maybe she has a number. Cell number? I don't know. I don't know. I'm going to my Bon, elle est là? Ok, déjà.
Bonjour.
The administrative guidelines. Okay. Hi, Julie.
Hello.
Hello, thank you. Thank you to be on time because we were a little bit early. So I'm very thankful that you are on time. So I will let the floor to the president of the jury, Corinne Snarbanis, Professor Corinne Snarbanis, who is going to.
Okay, thank you, Michael.
Start
Hello, everybody. We're very happy to be here. I want to say hello to the big public. Everybody came to support you, Stella. Wonderful. It's always a very important moment in, your life today, your scientific life, at least, I hope. And I'm very happy to open up this PhD defense. I just want to give the floor 1 minute so that everybody could introduce themselves really briefly, just that everybody knows who we're talking to. And then we'll begin. I think you have 40 minutes to present your work, and as usual, we will discuss. Okay? Okay, please. Maybe we have colleagues online. If you would like to begin with just saying hello and where you are. Maybe Madame Joss, yeah.
Sure. So hello, so I'm Julie Joss. I'm a research director at INRIA, located in Montpellier, and I lead an INRIA INSERM team named Premedical.
Thank you. Madame Rita,
Anastasia.
Anastasia.
Yeah, everyone. I am Anastasia Grithara. I'm a researcher in the National Center for Scientific Research, Demokritos, which is the biggest research center in Greece, in Athens. My background is on machine learning and AI, and I'm the head of a group on biomedical and health informatics team. Really nice to be here.
Thank you. Please.
Hello, everybody. My name is Francesco Salvo. I am a professor of pharmacology at the University of Bordeaux and the chief of the Regional Pharmacovigilance Center of Bordeaux.
Hello, I'm Dimitris Kalles. I'm a professor of AI with Hellenic Open University and director of the Data Science and Machine Learning program there.
Hello, my name is Pantelis Natsiavas. I am a researcher in the Center for Research and Technology Hellas based in Thessaloniki, and I am the head of the eHealth lab.
Hello, I'm Charles Assad. I'm a junior professor in CERN. I'm specialized in causal inference and leading a team specialized also in causal inference at l'Institut Pierre-Louis d'Épidémiologie et de Santé Publique. Thank you.
Yes, I'm Marcus Roland, so I'm the supervisor of the Stellar thesis, research director at INSERM, and co-supervisor because the other supervisor, important one, is Pantelis.
Okay. And just I'm Professor Banis, I'm a nephrologist, professor in nephrology. I'm involved in digital health, but in a very different way compared to what you're doing, but very interested in toxic acute renal failure, as you know, And that's one of the reasons why I'm here today. So we are really happy to welcome you today, Stella, and we listen to you. If you'd like to begin your presentation.
Yes, thank you very much. I will just move the swinging for now on the screen. Okay. So, hello everyone. I'd like to thank you, the members of the jury, for accepting to reviewing my work, and of course my supervisors for supporting me these 3 years of PhD. The title of the PhD I will present now, today is the use of artificial intelligence for the analysis of potential pharmacovigilance signals upon real-world data. I will start by introducing the context of this work. I will start on how artificial intelligence shapes the future of medicine. So based on National Institute of Health and Care Research of UK, AI in medicine can be classified in 4 categories, starting with predictive AI, which predicts future events based on historical data, diagnostic AI, or therapeutic AI, which analyzes complex data in different medical events, generative AI, which produce a variety of content, text, audio, etc., and assistive AI, which augments AI and human knowledge in medical tasks. Next, I will introduce pharmacovigilance. Pharmacovigilance is the science and activities relating to the detection, assessment, understanding, prevention of adverse effects of any other medicine-related problem. And adverse drug reaction is a response to a medicinal product which is noxious and unintended. And with response, in this context, we mean that a causal relationship between the medicinal product and the adverse events is at least a reasonable possibility. Based on the recent literature, adverse drug reactions are the 5 to 7% of emergency visits and prolonged hospital stays. The last pillar of this work is real-world data and real-world evidence. Real-world data are data relating to patient health status or the delivery of healthcare routinely collecting from a variety of sources. And real-world evidence is the clinical evidence about the usage and potential benefits or risk of a medical product derived from the analysis of real-world data. Advantages of real-world data is that they capture a broader patient population than randomized controlled trials. And key challenges is the data quality, as the data are collected not for a specific purpose. They may include a lot of noise or biases, and also the privacy concerns, because when we are talking about real-world data, most of the times we are talking about sensitive data that they need strong regulation to be protected. The research questions that we aim to answer in this work are 3, starting with pharmacovigilance, where traditional PV methods face challenges such as underreporting, reporting biases, and the limited sample sizes. And AI can offer opportunities to improve PV methods through large-scale data analysis that uncover complex patterns. But AI, it didn't test in full scale until now in the field of pharmacovigilance. So our first question is, how can AI contribute to the enhancement of pharmacovigilance procedures? The next question is regarding real-world data, which, as we said before, provide a vast source of patient-level information for ADR detection. Most of the studies working on real-world data and pharmacovigilance, they are working with unstructured real-world data with, for example, clinical notes, which can include human biases or subjectivity. On the other hand, structured real-world data, like laboratory tests or medication, follow standardized procedures to be collected, which reduce human biases in an extent. But also, structured real-world data include other types of biases like time biases, etc. So our second question is, how can structured real-world data enhance NDR detection with addressing challenges such as accuracy and bias in decision-making? The last question is about AI field. So given the regulatory and ethical considerations surrounding AI in healthcare and more specifically in PV in our case, questions arise about the practical implementation of these technologies. So the last question that we aim to answer is, how can AI models be made interpretable and transparent for regulatory bodies and healthcare professionals? In order to answer the first question, we apply a scoping review titled Applying AI to Structured Real-World Data for Pharmacovigilance Purposes. And the aim of this review is to identify and characterize the current research trends regarding the use of AI on structured real-world data for pharmacovigilance, and also highlight the relevant gaps. The query is structured based on the synonyms of pharmacovigilance, structured real-world data, and artificial intelligence. And the framework that they use for this study called PRISMA framework and help us to conclude to the 36 included studies that we analyze further. In the results, the study's characteristics that included published in 2021 from United States and from academia and research organisms, which in our case can be a bias, can call a biased result because we use only PubMed literature database to search, to collect our studies. Our studies are further classified based on machine learning methodologies that use real-world data types and PV processes. So from this classification, we end up that most of the studies are using classification models with random forest algorithm, and they evaluate the results with area under the curve, ROC curve, and the type of real-world data that they use mostly are electronic health records, and the field that is mostly examined in PV, it's ADR detection. To evaluate our results, we follow a series of qualitative evaluation, starting with code availability in public registries. As we can see from the results, most of the studies are not published in, they don't have published their code. Clinical validation, in most of the studies, the AI models didn't test it in clinical environments. And the data preprocessing pipelines, in more than half of the studies, the data preprocessing pipelines of real-world data, they are not detailed mentioned. The last metric that we use for our work is regarding trustworthiness of AI. For that, we use Future AI initiative. Future AI comes for fairness, universality, traceability, usability, robustness, and explainability. And as it's shown from our results, there is a lack of usability testing in our results and also lack of explainability of AI models. The strengths of this work is that we include a considerable number of studies, 36 from 2010 to 2023, where the last review in this field include only 7 studies from 2015 to 2021. And key limitation is that we didn't use any kind of meta-analysis in the quantitative results because of the variation of articles methodology. This work is further enriched with studies after 2024, which highlight that there is an increased interest in graph AI methods in ADR detection field. And we also published this work in JAMA Journal. The gaps that are highlighted from scoping review is the lack of federated data networks, the lack of detailed analysis of real-world data preprocessing pipelines, lack of explainability of AI, clinical validation, and models transferability. And as we said when we described the adverse drug reaction, causality is a very important thing that can enhance pharmacovigilance. So our hypothesis is whether causal machine learning can fill some of these gaps and provide possible solutions, like in real-world data preprocessing pipelines, in explainability, and clinical validation. So this drive us to reframe the first research question on how can causality be applied to structure real-world data to improve pharmacovigilance. In order to answer that, we start by examining the literature on what extent causal machine learning application applied in healthcare. Based on Pirger et al., causal machine learning could enhance clinical decision support systems by giving the opportunity to healthcare professionals to make personalized predictions on how a patient's health state alters under different treatment options. And we detect studies in this field in medical imaging and in pharmacology field. We further examine in what extent causality and machine learning are integrated in pharmacovigilance field, and we classify our results based on the data that they are used. Starting with FHIR's data, with individual case safety reports come from US Food and Drug Administration. In this field, we detect 2 studies, one that integrates causality assessment with classification modeling, and one uses InfraBird for applied in nodes in FHIR's data in real text. Next is the application of causality and machine learning in biological and chemical data. Here we detect one study applied structural learning with classification modeling. And lastly is the studies that work on electronic health records, which are more related with our study because they are using real-world data. Here we detect work using targeted maximum likelihood estimation and one work on causal discovery field. But these studies we are really interesting and give us the initiative to search more in this field, but they are not aligned with the needs of our work, which insist on more individual level analysis of patients. So we, in order to answer this question completely, we further search how causal inference can be applied on real world data and also support the needs of the goals of our study. Starting this procedure with designing the causal question. For that, Heron and Robbins propose a target trial approach. In this approach, we're trying to mimic randomized controlled trial structure on observational data. And it has 5 core components, starting with eligibility criteria, who is eligible to participate in the work, treatment strategies, which are the treatments that we want to compare, assignment procedures. Even if we cannot have randomization on observational data, we have to mimic randomization by adjusting for possible confounders. Follow-up period is the period that we want to study the reaction of drug, and finally the outcome that we want to measure. Next step is the adjustment for non-causal paths. In this step, we need to design the causal graph, structural causal model, or directed acute graph, known as DAG, in order to detect the possible confounders of our work, and further continue with the matching procedure in order to create a similar distribution of covariates between the 2 treatments that we want to compare for the patients. The last step is the treatment effect calculation. So in order to apply causal inference on observational data, we first have to make strong assumptions, starting with ignorability, which states among the patients with the same values of covariates, we suppose that the treatment has been randomly assigned. Then the next assumption is positivity or overlap, which states that for every set of values for covariates, treatment assignment was not deterministic. And the final assumption is consistency, which states that the observed outcome of a patient under a specific treatment is the same with the potential outcome of this patient under that treatment. Next, to calculate the treatment effect, we use conditional average treatment effect, which quantifies the expected treatment effect of subgroup of patients with covariates, and individual treatment effect, which describes the difference between an individual's outcome when receiving the treatment versus not receiving the treatment. And the final is a sensitivity analysis, which measures the robustness of our average treatment effect models. This brings us to the second research question on how suitable are structured real-world data for causal machine learning using pharmacovigilance, and how can enhance ADR detection while addressing challenges such as accuracy and bias in decision making. In order to do that, we select drug-induced acute kidney injury case study. Acute kidney injury or acute kidney failure is a sudden episode of kidney damage or kidney failure that happens within a few hours or a few days. And based on research studies, drug-induced acute kidney injury is estimated from 19 to 26% of all acute cases in hospitalized patients. And in order to detect patients with acute kidney injury, we use KDIGO guidelines, which range the different acute kidney injury states from 0 to 3. The real-world data that we use in our work is electronic health records. Electronic health records are real-time patient-centered records that provide immediate and secure information to authorized users. EHRs typically contain a patient's medical history, diagnosis and treatment, medication, allergies, immunization, radiology images, and laboratory results. EHRs also include diverse population and cases of drug misuse or drug overdose. In order to detect the suspect drugs to acute kidney injury outcome, we select two studies, starting with a recent study of Fernandez-Lanez of 2025, which includes, which ranks the suspect drugs to acute kidney injury outcome from strong effect to limited effect. And also an older one comes from Ryan et al., which includes suspect drugs to acute kidney injury outcome, but also negative drugs that are known based on this study that are not related with acute kidney injury outcome. The database that we query is Mimic4 database. It's a free available database including electronic health records. And the drugs that we managed to find an adequate number of patients that can produce evidence for our work is 8 suspect drugs: ibuprofen, ketorolac, vancomycin, lisinopril, furosemide, pantoprazole, and omeprazole. And as negative drugs, we have simethicone, prochlorperazine, and lantulus. For the 24 datasets that are created, we include in each dataset patients with acute kidney injury outcome and patients without acute kidney injury outcome. And each one of these datasets, the patients with acute kidney injury outcome are less than the patients with non-acute kidney injury outcome. The covariates of the patients, the variables of the patients that included are concomitant drugs, which are drugs that co-administer with the suspect or the negative control drug during the period that we study, blood pressure, laboratory test, weight, gender, Charlson comorbidities, and age. And the number of variables in the 24 datasets range from 101 to 502 and is related to the number of concomitant drugs in each dataset. So, as we said before, we include acute kidney injury patients, which the interesting information starts with the drug administration and ends with the acute kidney injury outcome. And non-acute kidney injury patients, we start with the drug administration and we end with the end of the last dose of the drug that we are investigating. In the beginning of the drug exposure, we include the age of the patients and the gender. And during the drug administration, we include the minimum weight, the mean of blood pressure, the number of Charlson comorbidities are included, and the number of concomitant drugs. The laboratory tests are represented in two ways, as mean values and as first and last value. First value in the beginning of the drug administration and last value when the acute outcome happens or in the end of the drug administration. And this brings us to the last research question of this work, which is referring to how can causal AI models be made interpretable and transparent for regulatory bodies and healthcare professionals. So pharmacovigilance studies are prone to biases such as publication biases, data dredging, and selective reporting that can inflate false positive and undermine reproducibility in drug safety research. So our hypothesis is that a protocol-based causal machine learning framework that emulates a target trial can reduce biases and improve the accuracy and reproducibility of ADR detection in electronic health records. To do that, we select to work with the principal protocol, which is a stepwise process that helps guide key decisions in study design and data analysis for non-interventional studies, and aiming to produce reliable and reproducible results. So the first step of this protocol is to formulate the causal question via specify the target trial protocol of that we want to use. In our case study, we work with PICOT from protocol. PICOT comes from population intervention control outcome and time, and the produced research question is among hospitalized patients, what is the causal effect of receiving a suspected nephrotoxic drug versus a negative control drug on the incidence of ACI occurring after the first day of drug administration. The second step of this protocol is the emulation design. In this step, we provide a detailed description of the data preprocessing pipeline that we follow, and also we design the causal graphs. In order to design the causal graph, we start by reviewing the existing literature on drug-induced acute kidney injury and collect the type of variables that mostly included in these studies. In the next step, we use statistical testing to investigate in what extent the variables are correlated with the reaction, the suspect drug, and the reaction that we want to study. So we end up to create a non-directed graph based on the statistical testing, and then we further review the literature, this time to select the direction that of each node based on the variable between the variables and also with the drug and the event, the reaction. In the last step, in the graph that is created from the we discuss the graph that is created from the literature with a nephrologist and more specifically with Professor Albanese. And we discuss in what extent some ages can be changed based on her expertise in the field. In the third step, the precision diagnostic, we first have to do the assumptions that we mentioned earlier, and we calculate the minimum detectable relative risk. The minimum detectable relative risk quantifies in what extent the data set, the cohort of patients are able to detect small effects. So we, as a threshold to MDRR, we select 1.25, which comes from the work of Ryan et al. And our results, in our results, we have MDRR for all of our data sets less than 1.12, which indicates that our data sets are able to calculate small effects. In the step 4, in bias medication, we follow 3 different types of sensitivity analysis. One, based on the representation of laboratory values. As we said, we represent laboratory values as mean values and also as first and last value. Second, with different confounder sets, during the discussion of the graphs that are created from the literature with the nephrologist, the literature and the nephrologist's opinion differs in some nodes, some connection of the nodes. So for these differences, we calculate average treatment effect in these 2 different cases. And the last type of sensitivity analysis that we use is regarding normalization of laboratory tests and vital signs. So we represent values as laboratory tests and vital signs as raw values and also as normalized. Next, for the last step of this protocol is the inferential analysis. So we apply matching with propensity score matching, and we evaluate the results with standardized mean difference. Standardized mean difference indicates if the smallest standardized mean difference implies the best similarity in the distribution of covariates between the treatments that we compare. And we use the models that perform better to SMD2X learner, which is one of the architecture of the causal machine learning methods that we use to calculate CATE. Next, in this architecture, we select to use 2 classifiers and 2 regressors. Next, in S-learner architecture, we use 1 classifier, in T-learner 2 classifiers, and in TML 3 regressors. Here you can see the bigger picture of the models that are tested for their performance in causal machine learning architectures, and also the propensity score matrix and the models that are testing in the propensity score that are testing for the best propensity score matching performance according to standardized mean difference. So the last step in inferential analysis is the evaluation metrics. We evaluate our results in 2 ways, starting with conditional average treatment effect. So in this evaluation methodology, we evaluate the confidence intervals width. So we set as valid the CATE results that have confidence interval width less than 0.85. And also we examine the alignment of the ranking of the effect produced from CATE with the study of Fernandez Laneza. As we said, when we introduced the study, which includes suspect drugs for acute kidney injury, Fernandez Laneza study ranked the effect of the suspect drugs to acute kidney injury outcome from strong effect to limited. So we're trying to rank our results with CATE and see what are the similarities and the differences with this study. And the second way that we select to evaluate our results is with individual treatment effect. In this step, we classify the patients to protective effects, patients with individual treatment effect less than minus 0.1, no effect, patients with individual treatment effect range from minus 0.1 to 0.1, and as patients with adverse effect, the patients where individual treatment effect is more than 0.1. With this, for these categories, we examine what are the mean values of the different categories in laboratory tests, vital signs, and demographic results. And we apply statistical testing to see if the difference in means in the variables of patients is statistically significant. And we further, if this happens, we further examine the alignment of this significance with the existing literature. Starting the results with causal graphs for 4 out of 8 drugs, suspect drugs, the graph that proposed from the literature is slightly different from the graph that proposed from the nephrologist, and for 4 the literature the rest 4 drugs, literature and nephrologists agree in the same structure. The last the number of the created graphs is 12 that are tested in this study. One really interesting difference between the two the ways that nephrologists and literature propose the graph is that in the literature there are studies that that suggest that the allopurinol is directly connected with acute kidney injury outcome. And from nephrologist's view, the allopurinol is not directly connecting with the acute kidney injury outcome. In the matching results, we can see that most of the drugs, the suspect drugs, in combination with the control ones, they have optimal standardized difference in mean, which is less than 0.2. And for 3 out of 8 drugs, the standardized difference in mean it's way more than optimal. It's range from 0.3 to 0.5, which indicates possible confounding, or that the models that are used for matching couldn't fit well with our data. Starting with the CATE results, as we said, the first evaluation that we select to do in the CATE results is through causal confidence intervals which is less than 0.85. From these results, we conclude that most of the valid CATE values meet in the ibuprofen suspect drug, with combination with simethicone control drug. From nephrologist's view of structure of causal graph, the laboratory values representation didn't play any important role in these results, and from S-learner causal machine learning methodology. We further examine our results based on the significance of confidence intervals. So confidence intervals are significant in this study if they are not include a 0 in the range of values. And in our results shown that only 4 out of 8 drugs present significant results according to confidence intervals. Again, the most significant results are included in ibuprofen drug in combination with prochlorperazine negative control drug. Literature has slightly more significant results, the graph that are created from literature. And here, the most significant difference is the way that the laboratory values are represented in the data. So we have most significant results when the laboratory values are represented as first and last value. The causal machine learning method that includes the most valid, the most significant results is XLearner, and the negative Kate results are slightly more than the positive ones. In the last step of the evaluation of Kate, here we compare the results coming from the literature. Here is the ranking of the drugs from the literature from very strong to limited. In this study, the drugs are ranked based on analysis of individual case safety reports databases and also based on the existing literature. And here is the ranking of our results based on Kate values. So here in the ranking, we include the Kate results that are significant, so they are not include zero in the confidence intervals for the drugs that exist, because as we said, only 4 drugs have significant results based on confidence interval. And the most the biggest difference in this ranking comes for furosemide drug and ketorolac, which are not aligned at all with the ranking provided from the existing literature. In the individual treatment effect evaluation, we here in the laboratory test demographics and vital signs are the variables that have significant statistical significance when we compare the patients based on individual treatment effect for the 3 categories of patients: the patients with no effect, the patients with protective effect, and patients with adverse effect. And we indicate with this arrow the results that are significant increase in the case of adverse of the patients categorized as adverse effects, and decrease the patients with categorized as adverse effects. And we further search the existing literature for how much the literature support our results. For these 2 drugs, we can tell that the literature, our literature align, or our results align with the existing literature in a significant extent. So the strengths of this work is that we develop a reusable, detailed causal machine learning framework for drug safety evaluation based on principal protocol, covering all stages from data extraction to individualized treatment effect estimation. And we apply this framework in 8 real-world cases, emphasizing the importance of involving clinical experts, such as the nephrologist, and in the analysis process. The limitation is that our framework is applied only in Mimic4 database, which minimizes the generalization of our work, and we didn't include information like patient socioeconomic status and drug dosing in our data. Another limitation is that we collaborate only with one nephrologist and one hospital, and we need more clinical validators in the pipeline of our framework, of our work. So our study proposed a principal reproducible analytic framework using structured electronic records data to improve causal ADR detection in pharmacovigilance. We're trying to reduce the confounding by creating causal graph informed both from literature and from clinical experts, and we test multiple control drugs. Our study concludes that no single causal machine learning method is sufficient, and the model choice and the data characteristics critically affect the outcomes of the effect. And based on individual treatment effect results, we noticed that drugs with stronger effects, that the ones that we present here, ibuprofen and ketorolac, better align with the existing literature. So we assume that better described from our proposed framework. And finally, the subgroup analysis by demographics and lab values reveal patient-specific effects, many of which are consistent with mechanistic sites from literature, which underscores also the causal machine learning's value in personalized pharmacovigilance. Concluding this work, our contribution are that we provide the synthesis of the current landscape of AI-driven pharmacovigilance using structured real-world data, and we're identifying the gaps, the strengths, and the future directions. We suggest how causal inference can be applied in pharmacovigilance field. We produce a detailed data preprocessing pipeline, or how electronic health records can be preprocessed to support ADR detection. And finally, we develop a complete causal machine learning framework analysis pipeline, starting from the data preprocessing to the calculation of the effect. Our limitations is that we didn't use any kind of metric to calculate the unknown confounding in our work. Even if there are different AI methods like multimodal AI, multitask AI, that can also fill the gaps that we highlight in this work, we didn't test them. We select only to test causal machine learning. And we use Mimic4 database and drug-induced acute kidney injury case study, which limits the generalization and the use of our framework in different ADR detection cases. And even if we have the help of nephrologists in the creation of the causal graph and the literature to validate the last results coming from individual treatment effect, we didn't test our framework in clinical environments. And finally, we didn't apply any kind of sustainability. We didn't use any kind of sustainability analysis metric to quantify the environmental footprint of causal machine learning models or real-world data storing in our study. The challenges that we face is in the finding data for external validation. So electronic health records are sensitive data that and strong regulation protect them, especially in European Union. So to get access to this kind of data is a time-consuming procedure. And we didn't manage to have access to the data in the early stage in order to test further our framework. And also, a significant amount of time spent it in building fundamental expertise in causal machine learning, and because it's a new and complex domain, and to find out how we can apply in the pharmacovigilance field. Our perspective is to enrich the causal machine learning framework that introduced here in PV and electronic health records by testing different ways of data representation, like a self-control case series, and integration of our data with other types of data, like biological data, to test different causal discovery methods that exist in literature to enrich our causal graphs. And the recent studies that propose also different loss functions for selecting the most optimal machine learning model for the causal machine learning architectures that we are going to use and test in our framework. Also, we are thinking of testing more causal machine learning methodologies like neural methods or ensemble-based causal machine learning architectures. And finally, to examine the combination of causal machine learning with explainable AI methods and further investigate how this can enrich causality and pharmacovigilance. Thank you very much.
Thank you very much, Stella, for this nice presentation, perfectly on time. I think we now can open to the floor for discussion. And maybe we'd like to begin with Professor Calles. You'd like to...
Thank you. Well, I've made a couple of notes, and always, listening or attending the presentation gives you new insights and new ideas about what you would like to ask. There's, on a comparison with the text. So I'd like to start with the question on the variability of the pipelines. This pipeline that you presented is Stella's idea of how things will work. Do you have any concerns about, other professionals, either from medical informatics or from the medical profession, being happy with this pipeline or finding out what they will try maybe to rearrange in order perhaps to get the same results, but with more confidence in how they will present these steps? So a comment on the steps of this process.
Okay, thank you very much for your question. It was a big question also when I first started to work on how causal machine learning can be applied in ADR detection, because it's very new in the field. So in order to have a pipeline that is that I can feel secure to using in this context, I select to work with the principal protocol because it's a protocol published in the study is published in 2024, and it's a proposition of how you can use causal inference on observational data. So I didn't, because the field that I'm working is multidisciplinary, and I have mostly expertise in AI field, in order to not get out of the lines, but do something that can lead to completely wrong results. I decided to work to this principal protocol, and it's a really detailed protocol of how data preprocessing is gonna be made. So I think that it covers at least the reproducibility of the results in one extent.
Right. Can I have a follow-up question and then maybe give the floor to the rest of it? So my next question, it also has to do again with the pipelines, is what is, let's say people agree that pharmacovigilance can be done with pipelines like this one. What is the mixture of medical experts, medical doctors you expect to find in these pipelines? I mean, do you expect to only see them at the end evaluating the clinical results? Do you also expect to see them at the data collection stage, ensuring, let's say, data quality? How do you vision these people involved in this pipeline?
Yeah, this is the most important thing in the field that I decided to work for my PhD, because clinicians are mandatory in every step of the work, from the selecting the data, which variables are more important according to the case study that we work on, and also the phase that we include them in during the causal graph creation. It's for me, if you ask me, it's mandatory to have them also in this stage. And then, of course, in the end of the evaluation, the last step when you do the inferential analysis, because in pharmacovigilance, it's difficult to have a ground truth when you're trying to answer the question of with average treatment effect. You may know that there are average treatment effects that are in the literature, but this is not quantified in a way that you can have it as a ground truth during your work. So the last step, clinicians are mandatory more than in every step, but in the last step, they have to evaluate the results. Otherwise, you cannot support that you have a result in this field if they are not validate the last step of this work.
I might come back later. So
Okay. Thank you. So please, Professor Sabo, would you like to go on?
Okay, yes, thank you very much. So, well, first of all, congratulations, because it was a huge work that you did. And in particular, I like your limitation slide, in which you say, okay, we have problem with that access. We have problem with appropriation of how we can do causal machine learning in pharmacovigilance. And even after 25 years of experience in that field specifically, I am struggling with that. So it's quite okay that you did it in 3 years. It's very good. So also your slides were quite clear from pedagogical point of view. I understood a lot. So thank you very much. It was a pleasure also to read that. So also thank you to your director to invite me to do that. And I think that you are moving in the right direction, in the sense that pharmacovigilance classically was built up in a sort of solidarity model, in which every part of the world, every clinician of the world, tried to send to system cases of potential adverse drug reactions, and then mutualizing that, we tried to understand if there was a causal or not. It was, I would say, the first phase of pharmacovigilance up to 90s globally. Thereafter, step by step, we try to prioritize from cases, using some techniques to push up some events in order to go through in detail, named the disproportionality analysis that you also said in your thesis. And now we are in a moment in which we try to use real world healthcare data in order to have signals that we can come back thereafter to the clinician to evaluate. So I think that you are in the right path in that sense. So I made already my comments on the right part of the thesis. I really just would like to have some eventual starting with 2 or 3 questions, and then maybe make some interactive as Dimitris started to do that. At the beginning, I was really I thought that the causal machine learning was more at population level. But from your presentation, I think that you are more on the individual level.
Mm-hmm.
So when you speak about pipelines, it seems that with this kind of methods, we can help clinicians to say, okay, if you prescribe this medication in this patient, you have this risk of efficacy. Am I correct?
Yes. The individual treatment effect, which is the last evaluation that we did in our framework, is exactly for that. It's for to characterize which patients are more close to have the effect based on the values, laboratory tests, vital signs, age, and all the information of the patient. So yes, this is more helpful. We cite this as more helpful approach in the decision making when you have to apply the framework in clinical environments.
Okay, and this is quite nice, I think, because if we can come back to the original definition of pharmacovigilance, you fit with the risk reduction and the patient level. You fit with that, and it's very, very important on that point, because I think that go a little bit far from the title of your place. that you speak about signals, and then during your presentation, you speak more about ADRs. So something that we know that we have to manage more than finding something that we don't know.
Mhm.
And I would like to have your appreciation of that. So the difference between the positioning of this kind of methods in signal detection from one side and risk management, I would say, or risk reduction in the other side.
Yes, I was also questioning myself in the beginning because these terms are really difficult for me to understand in the first place. So after thinking about this and going further with the work, I think that our pipeline is more it's like complementary work in risk management, as you said, case, and not in revealing new signals. It's like if I can think about it as part of the pipeline in the future, I can say that we already know that this signal exists, and we are trying to have a causal approach before we are going to evaluate further from the clinicians and to go in the medical and 100% medical field, like the last step. It's a more mathematical approach in causality in the steps of risk management of the adverse drug reaction.
So, and in some sense, you try to help reason.
Yes,
That's-
exactly.
So it is a sort of also of risk prediction models or individually or not?
Yes, if we can describe it in the way you said, in the individual way, what are the characteristics of the patients that more able to have an adverse drug reaction? Yes, it's to quantify in the risk prediction of each one of the patients. Yes.
And after the last question for the first part, I would say, for the study design that you use, you can come back maybe to the slide 26.
Yes. Yes.
Because classically, in pharmacovigilance, in pharmacoepidemiology, say better, we have two kind of approach. Something that starts from the disease, and then we come back in order to understand the exposure. No, it's more on this. Sorry, I have a problem with the slide. Maybe. Oh, is that one?
Okay.
Great. So, or we start from the end. That it seems like what you did. So we start from the end and then we go back in order to understand if there is a difference in drug exposures between cases and controls, so people with non-AAT, or we start from the exposure and then look at what happened thereafter. So the exposure with to drugs, and then look to ibuprofen, for example, and then look at what happens thereafter. Here, I really would like to have that clarified, because you started from the disease, you prospect, so from disease that happened thereafter, and then you follow prospectively the exposure. So what is your study design? Is more a case control study design? Is more a what we call cohort design? Is something different that I don't manage because I'm not an expert of that. Is a self-control case? Is really, please, self-control of course, but
yes. Okay. So yes, it's not really clear how I detect the patients in my case. So my approach is to search for the database with electronic health records for patients that took the suspect drugs that we are endorsed, or the negative control drugs, the drugs we have for comparing the treatments, and what if they have an active kidney injury outcome, which is measured in our case with KDIGO guidelines. So if the number change from 0 to 1, 2, 3 after 24 hours, or after 24 hours, or 7 days after the end of the drug exposure, we have a change in KDIGO guidelines. We have an active kidney injury outcome. So we say we tried to measure. We didn't take into account the acute kidney injury, the change of acute kidney injury state after the before 24 hours of the drug administration, only after 24 hours. And if we don't have after 24 hours till the end of the drug exposure an acute kidney injury outcome, we see 7 days after the last dose of the drug if there is an acute kidney injury asthma. So we include also these patients as acute patients in our cohort. Yes, and yes, and the difference for the non-acute patients, we just take the information from the drug administration till the last dose of the drug.
And so in the non-acute patients, you take also the patients that I would say don't have an acute kidney injury after 24 hours or not?
No, for this category of patients, I have only patients that I have KDIGO 0.
Okay, thank you. Thank you very much.
Thank you.
Thank you. We'd like to hear Madame Joss, if you'd like to go on with your comments or questions, please.
Yes. So first, I would like to congratulate you for your work. It's a quite impressive and a large amount of work. And it was very clean in the sense that you proceed to all the steps to try to emulate the trials with a lot of care, and you try to do also a lot of sensitivity analysis, and you have tested a lot of models. So congratulations for that. Then I have a few remarks and a few questions. So first, maybe on the first part of your work, where you have reviewed all the AI models used in pharmacology. Well, it was you have mentioned that a lot of studies do not have code availabilities, and it's difficult to reproduce and so on. So in fact, it's a sad in some sense, but we still are in the process where there are a lot of papers on using AIs for medical data, but very few implementations, very few deployment. Yes, a bit of too many papers. So it was it's a bit sad to see this, but it's still the case. Then I have a few technical questions, so things I think I did not get. So first, maybe it's a matter of terminology, but I was just trying to check again in your document, but I could not find it. So can you tell me again, what do you mean when you speak about both conditional average treatment effect and individual treatment effect? Just for...
Yes. With conditional average treatment effect, I calculate the effect in each one of the different combination of suspect and control drug for each one of the 24 datasets, the overall effect. And in individual treatment effect, I calculate with the same models. I mean, the individual treatment effect is produced from the Kate is produced from the average of individual treatment effect. So then I went to each one of the effects of the patients to further see what are the characteristics and what classify them as adverse effect or protective effect or no effect patients.
Yes, so maybe it's for me, like what you first mentioned as your conditional average treatment effect is more like an average treatment effect, but because you have done like 24 studies, you have one for each study, if I am correct. But just my point is that often people use individual treatment effect, but really in causal inference, it's really impossible to be at the individual level. Even you can be conditional. Conditional is in the sense that you will say like, okay, the effect of the treatment depends on some features. So you just look at interaction between treatment and patient's characteristics, basically. And this is conditional. So basically we are able, like in statistics often, you are able to draw conclusions only for a subgroup of people. It's extremely difficult if you want to say something for me, let's say, You can just say, okay, I can give you like, okay, the effect of your treatment for you, it's not really for you, but it will be for a group of people that really looks like you, at least on all the features that we have, So maybe it's a terminology things, but to have really individual effect, like at the individual level, like the assumptions are extremely strong. So for instance, you need to have like a conditional independence of your potential outcomes given the features, which is extremely strong as well. This is just a remark, but often like in many papers, people speak, when they speak about ITs, is more like conditional effect. And I think it's what you do at the end of the day, because you look at the different features. Is it this?
Actually, yes, based on your description, yes, this fits better of my work. Yes, that's true. I mean,
Yeah, it's just like, in a way, we hope for personalization and for personalized medicine, but we are still a bit far in some sense, and to be at the individual level is like more difficult. It's really like by groups of patients with some features. But it was just to be sure, but I think it is this. And then I'm wondering, how did you build your confidence intervals? Because you mentioned like a threshold, like you mentioned the length of, if the length of confidence interval is below 0.85. So I just didn't get how did you build your intervals and how did you select this number?
okay, yes, you're completely right. This is so the truth is that I was searching for a way to evaluate the gait, and so I use a bootstrap method to calculate confidence intervals. And after I produce the confidence intervals, I've seen how the values range, and I select after what are the values that I can call valid for my work or not, because I check that in 0.85 is the max range of the values that are significant, that have significant confidence intervals, that don't include zero in their prediction. And it's very important to know if my results include zero, because I have to know if the drug is closer to classified as with a positive effect or with a negative one. So then after I realize that this is the biggest range of values, I select this as cutoff to further evaluate all my results.
Okay, get it. And then maybe a last on these technical clarifications. So at the end, you have tested many models to compute your effects, like X-learner, double machine learning, and so on. And within each class of models, you have used a lot of different ways to do your regression or your classifications with random forest or just logistic regression and so on. And so, can you tell me a bit, how do you decide, like for you, what is a good method or not? So for instance, at some point you say, oh, okay, X learner may be a bit more satisfactory.
If I understand correctly, you ask me how I select the machine learning model in the causal machine learning architecture,
No,
right?
it's more like within the causal machine learning models. You have different choice because you can do X learner, S learner, and so on. And so, what are your, maybe I didn't get like your recommendation on, okay, I have S learner, T learner, blah, blah, blah. Inside, I have a lot of things. And so, you test everything. And for you, how do you decide, okay, at the end, like which one you will trust more, basically?
Yeah, I mean, my results state that we didn't actually select one model that can be trusted, because the results differs. I mean, each model have different different behavior in every drug. So that's why we use the different evaluation metrics. First, we're trying to select as ground truth the ranking, compare the CATE results with the ranking coming from the literature. So we say we're trying to find the best balance between the domain knowledge and the validity of our results based on confidence intervals, based on the confidence intervals evaluation. So that's why in my results here, I tried to select first the models that has significant results, and then in the next for the drugs that I didn't have significant results, I select the models that gives me the smallest confidence intervals. So there's no one good causal machine learning model for each case. I mean, this is how we select actually the causal machine learning. But this doesn't mean that there are also other models in these cases that create significant results. We just select here to go, for example, in case of ibuprofen, to go with the result that has the highest effect, as in the literature, in the ranking, also has the strongest effect.
Yeah, so I completely agree that at least trying to have a kind of ground truth grounded in the literature can be good to help you a bit. Even though in the literature the populations can be different, and when the populations can be different, then the effect can be different as well. And so like the ranking maybe can be different in one population and in another one, like because if the effect of the treatment depends on the characteristic of the patients and the people are different in two studies, then you can see some differences. So you do not need, you do not need necessarily to find the same result or the same thing. So, it's a bit, that's why it's a bit difficult. It's the same thing like in observational studies. You do not need to retrieve the result from a randomized control trial neither, because studies can be different. So this is for me, a good approach, but also which has some limitations. Then my question was because I was a bit afraid of like when you mentioned like, okay, I select when I have the smallest confidence interval, let's say. This, I don't know if this is, maybe there is a large variability. It's difficult. So this is a difficult question because in causal machine learning, you have a lot of kind of causal machine learning methods, a lot of machine learning models that you put inside. Often, like there are a lot of qualitative appreciation, so people can display all the results from all the models, so which is huge. And then basically it's very qualitative appreciation in the sense that, okay, if all the models really say the kind of the same thing, there is a positive or negative effect, then we may think that it's an evidence to show that it's positive or negative. Because it's very difficult to know which one is a good one, because you don't know like how the features are, what are the relationship between variables. Each method has its own regime of predilection, its own theoretical guarantees. So often it's really like a lot of kind of qualitative aggregation. So my point is that in a way, I just prefer like to see a lot of the results of all the methods that you have tested to decide, like a lot of box plots just to see if, like, okay, all the methods on this data seems to agree. Because we know that some of them are more variable, some of them are more greedy in terms of data, and so on. Okay, so thank you. And maybe just a last question. It was just maybe in terms of outcome. So at the beginning, when I read your work, I was expecting to see, it was maybe naive, but to see more like, okay, I'm going to study like impact of some treatment or some combinations on a lot of outcomes, because we were speaking of adverse events. So on kind of multivariate outcomes. And is it not something that you were interested in or you can do in your data, with your data? Because here basically you just look at one univariate outcome in this study.
You mean if I was planning to add more adverse events than the acute kidney injury outcome that I studied, correct?
Yes.
Yeah. For these specific drugs, I mean, the way that the study was designed was to examine only the acute kidney injury as an adverse event. So we didn't further plan to have multiple outcomes for a different adverse event. Yes, I mean the whole study is planned to study only one.
Because in this framework, I guess it can make sense. You can have like efficiency of a drug, but then there are many adverse events that can occur. Maybe you can even have a hierarchy on these adverse events. So I think as a kind of a follow-up work, it can be interesting if you have the information and the data, because you can study the impact of the treatment on multiple outcomes and taking, I don't know, a hierarchy of adverse event. Or maybe it doesn't make sense. It was just a.
It was just tricky to see how I'm gonna detect, how I'm gonna name as a possible adverse event the information coming from electronic health records. So to go with an unsupervised approach, I mean, I think you need more help from a clinician to tell you what are the outcomes that you're gonna search in the data, because the data are massive in the electronic health records, and the outcomes are, the ICD codes are too many for each one of the patients. So you need to have clinical guidance to do, I assume, to do this kind of study.
Yeah, completely. Yeah, you need to have a bit of a priori knowledge, maybe. Or maybe a last one, just because I see this slide, sorry. Then you have performed like 24 studies, and do you see a way maybe to link this? Or, because it's really like you study like each combination of your control and your suspect drugs. So my idea is that, like, okay, do you know that there are some links between these suspect drugs so that you expect similar behaviors? Like in terms of the properties of the drugs and these kind of things, this should be included as kind of a priori knowledge or a kind of cluster analysis, if you see my points.
Yes, this is a very good question, because I also question myself for what are the control drugs. If the control drugs that I selected, they must have similar behavior with the suspect drug in order to say that you can matching the patients correctly. I mean, I selected to not further investigate that part. I think it's very interesting, and I'm planning to see further after the PhD, because it's very interesting. But I only select for this study to go with the negative control drugs that are proposed from the literature for comparing different methodologies, computational methodologies. But yes, I think it's a really interesting option for further investigation for the control drugs, at least.
Okay, so I think I am done for now. So thank you again for your work and all your work, especially as a sensitivity analysis part with different DAG, different features. It's great. So thank you.
Thank you. I would like now to give the floor to Professor Hassan.
Junior professor.
Professor.
First of all, yeah, thanks a lot for this presentation. I thought I enjoyed it. I learned a lot from it, even from the manuscript. I learned a lot from the pharmacovigilance domain. I didn't know anything about it before. And I agree with you that we need more causal inference in health, which is surprisingly not that present. And I think we need more PhDs like yours to pave the way for everybody ahead to use more and more of these methods in health. This is done. Now I will. The most important comment I have was already addressed, which is regarding the individual treatment effect. So good for me. That's also on the side. So in my questions, I will challenge you on the graphical aspect of the pipeline you proposed. Yeah. First of all, like on slide 40, you talked that you had two different graphs. Okay. I didn't quite understand how did you choose your last graph. I understood that you compared, you assumed each one of them, you tested both of them, but in some cases, like, okay, so one question, what they disagree about? Do they, for example, if you put both these graphs together, you get a union graph, do you get some cycles?
Okay, I know, wait, okay, so I'm sorry. You mean that merge the two?
Yeah, imagine, let's merge them together. Would you get some cycles? Do they disagree on the acyclicity? Or just on the absence or presence of an edge?
Absence or presence of an edge.
Okay.
This is the difference.
Okay, and they would not create some cyclic relations. Let's assume this. Let's assume that if you put them together, you will not get some cycles in the merge.
You cannot know that though
In advance, but if you get both, you can just put them together and check if there's cycles.
Yeah, I mean, I cannot answer you. I'm not fully sure, but the difference is in the absence or existence of edges.
Okay.
So I can tell you that in most of the cases, the literature has more edges, and then they eliminate or they question it from
That's
number.
like, for example, in the graph you showed, yes, there were one edge and then we removed it.
Yes.
So my question, I mean, of course, maybe you trust one graph from the other because of who gave you the graph. So maybe it's the expert acts on that. That's one way to do it. But let's assume we treat everybody equally. Okay, we don't know which is the best graph. What would be the most prudent thing to do in this case?
So actually, I will go to your presentation that you did in Linux, and you said that it's better to have more nodes than not having the edges. I'm sorry, yes, and that not having them. So
Exactly.
from that perspective, in order to be sure that I cover all the possible confounding that I can cover in a graph and all the structure, I will go for the one that has the most edges.
Okay, yeah, that's it. And here I feel, if I understand correctly, this graph, this specific graph, if I understand it correctly, it feels to me nothing changes. In terms of adjustment sets, whether you add this edge or not, there's absolutely no difference in so, which is perfect. So
Right.
for me, the nephrologist and the literature in these two cases, given this specific question you have, they totally disagree. Even though on this specific edge they don't disagree, this edge specifically, I think would have absolutely no incidence. So
You're right, in the confounders and there is no difference, but it's important from medical perspective.
yeah, from the description, not for the estimation part
Exactly.
of the causal effect, but from the qualitative understanding of the domain, yes, it can be very useful. My second question, did you try to test these graphs, each one of them? Like, not only these two. Each time you have two graphs, and there are methods that, I'm not talking about causal discovery. I'm talking about the testability of a graph, because each graph imply some conditional independencies. Okay? And so you can test these conditional independencies. First, did you do it? If not, it's fine, but do you have a reason not to do it?
No, I don't have a reason. It's just part of lack of knowledge I have in the field until now.
Which is fine. But that's one thing to consider. Sometimes, yeah, it would be nice to check how much the graph is compatible with the distribution. However, I totally understand sometimes when you have very complex data, mixed data, etc., you need a good tool to test them. You need a condition dependence tool. So if you are in a case where the data are very complex, etc., then testing it would make no sense. But it's worth trying if you are in a case where it's testable.
Yes, it's very interesting your point. Yes, I will be taking it on.
Okay, and one more thing also related to the same thing. So using a graph, you can get many adjustment sets. I didn't feel, maybe I missed it, but you didn't tell us how did you choose your adjustment set, because there can be many that are correct.
Mhm.
Right? I think
Yes,
in
no,
your manuscript, you talked briefly about this.
Yes, I have in the manuscript, I have the table
Yeah.
with the adjustment set I use in each one of
How
the,
did you choose it? If there's many? If there's many correct,
yes.
how do you,
It-
why did you choose this one and not that one?
There's no specific strategy. I just select one of the adjustment sets that proposed, but I didn't. There is no. I didn't know also that the selecting. No, I mean I didn't go so deep to check if there's different adjustment sets. What are the possible average treatment effect results if I change the adjustment set, but it's something that
But
I have to test.
But in your manuscript, or maybe it was, you were saying that we can get the minimum adjustment. You put like the causal analysis steps, and you're saying, yeah, we get the graph, and then there's many adjustment set, and we can find the smallest one or the minimal one.
Yes,
in the manuscript.
when I described the pipeline in chapter
Yeah.
3, yes, but I didn't,
didn't
follow it. No.
It's good that you didn't follow it, because actually the smallest is not the best.
Okay.
I mean, there's no smallest or largest means nothing finally. It has been recently proven that the graph itself is so informative, it can sometimes tell us which one is the best. And there are specific criteria for this, and they can improve your variance. And so that's another thing to take into account.
Yes,
for,
sure.
Yeah.
Yeah, very interesting. Okay.
Right.
Sure.
Again, about the application in nephrology, I'm not an expert. But I'm interested in the domain. And my question is the acyclicity assumption imposed on the expert? Like, did you tell the expert, I want a DAG, I want it to be acyclic, and you work together to get an acyclic graph? Or the acyclicity was just natural and it came without imposing this assumption? Why I'm saying this? Because while checking a bit online, and it's not reliable, and I'm not an expert again, but as I understood, like, for example, creatinine can cause hypertension, hypertension can affect creatinine, creatinine can affect urate, urate can affect creatinine. So it seems that it's a very complex domain. And I would like to know, I'm very interested to know if the acyclicity assumption holds, or is it actually an assumption you're imposing?
So the way that we discuss the causal graphs is that I create the initial graph based on the literature, where I knew in the beginning the theory about the acyclicity. So I was very specific, and then we didn't discuss it in a full extent. I just asked from professor to tell me if these edges are correct, or if these edges are supporting the expertise or not. So it didn't happen natural, and it didn't, I was biased when I create the initial graph, and then we didn't discuss about the acyclicity with it, but it's really interesting, that part, and it's really complicated for the knowledge I have until now in the field of causal graphs.
It's not just that. I think all of us working in the DAG-based causal inference, I'm not talking about potential outcome framework, but we are all tend to be biased to force the acyclicity assumption, and maybe we should not. Maybe we should be very careful and not to impose it on the experts. Okay.
Would it be a breach of protocol if I chip in with a question with that issue? Will it be? Go ahead. You know, coming back to my original question about scale, because you know I'm trained as an engineer, we're always interested in systems and scale. It looks to me, and I think that the discussion confirms it, that if you need to go through developing a drug yourself, taking it with a clinician, with every disease and substances that you think might be interesting, or if pharmacovigilance detection signal, then you are looking at a hugely expensive problem. So there is an enormous room for thinking about savings, about how to do that automatically. And doing that automatically will allow you to use what Sharon says about tools that, you know, how to put ensure that a certain syntax emerges and that syntax is correct, so you don't have to spend a lot of time yourself or with an expert developing that. Do you think that developing that part of the model has a fairly very expensive part of the process? I think it is, but
I mean, at least for me to review the literature and take all the domain knowledge and trying to create the graph, it was really time consuming. And I don't know in what extent I couldn't think, because I tried to find a way to maybe do it more systematically to see how I can gain time. And but even, for example, when I try to use the LLMs that are now trending, but it was really difficult for me that didn't have the domain knowledge. I'm in a computer science field. I don't know anything about nephrology and acute kidney injury to trying to select what is the valid, if what produced, for example, for an LLM, if the information that produced is correct or not. And I couldn't do it this systematically. I don't know in what extent, and I find it really difficult to produce, to automate the production of a graph without a clinician give. Because if you think about it in the extent of data, for example, an example that also I have in mind when I was designing the graph is that if you're trying to design a graph in a hospital in France and the same graph in a hospital in the United States, with the different population, with the bias included, it depends on the hospital that you choose, the habits of the patients. You can change completely the structure. So it's very difficult to think a way to automate this procedure.
thank you. And all of this was
It was very relevant. So, but you mentioned something at the end, which is not exactly a response to the question, but causal discovery. But it requires assumptions, and yeah, unfortunately, right now it doesn't work that well. But that can be a future solution for them. But anyway, so I will move on to my third question. As a computer scientist and a mathematician, I like to optimize things. And at some point, I saw a figure where you were saying that you have the query, then you get the causal graph, and then you have some assumptions, like ignorability, positivity, and consistency. Why is the graph is an assumption?
Yes.
Right?
Yes, you're right.
We should never forget this, and the graph is an assumption. And that's the beauty of it. At the end of the day, we can discuss it, we can put it. So it is an assumption. We should put them together. And when I said optimize things, I meant, given the graph, why you need ignorability? Either you have a set of variables and you assume you have ignorability, or you have a graph and you don't need an extra assumption, because the graph will tell you if ignorability is satisfied or not. If you assume that, of course, under the assumption that the graph is correct.
Yes, yeah, okay.
You see my point?
Yes, I see, I didn't think of
Allows you
it
to
this way.
get an adjustment set, but also the graph, when you find the most direct or simplest approach is to find a set of confounders that you adjust on them. You make your collision effect identifiable. And also related to this is more about the manuscript. You didn't talk about this in the presentation. And honestly, it's very small technical detail, which is not that important, but I think it's important to say it out loud. Do calculus, you talked about it. Yes. Very briefly. But the way it was written in the manuscript, the way I read it, maybe I'm wrong. It felt that the do calculus can help you get a do-free formula. But if it doesn't work, then we need to block some paths using propensity score, etc., etc. Is my interpretation of the text correct?
Yeah, I cannot clearly, I mean, I remember that mentioning briefly the do calculus part because I didn't want to go really into the theory and the theoretical part. Yes, no, yes, it was correct here.
And which is totally understandable that you didn't want to go in depth because you're assuming there's no unmeasured compounding, right?
Yes.
And so the do calculus is correct. You can use it, but it's not that useful compared to other simpler methods. But so my remark here, and it's something that I really would like to emphasize, and it's not a criticism whatsoever. I heard this many times. But the do calculus is actually, as far as I know, the most complete tool we have. If with the do calculus you cannot make it happen, then it's not possible to make it happen, except if you make more assumptions. So if your assumptions are just the graph, consistency, and positivity, then do calculus is the most complete thing you have. Nothing works if the do calculus tells you that you cannot get a do-free formula, your propensity score would not work, your S-learners would not work, nothing works, assuming the graph is correct. So that's very important. And why it's important, because why I'm so interested in your PhD, because again, you're paving the way for everybody ahead. And I think it's such a shame not to see people using such powerful tools that exist since 1994. And yeah, so I wanted to emphasize this. At some point, I had another question, but then I realized that there's no unobserved confounding. I think now I can let somebody.
Come back later.
Yeah.
Okay, thank you. So now we would like to turn to our colleague online. If you'd like, Mrs. Fritara, if you want to take some time to comment the work of Stella, please.
Yes, sure. Thank you. First of all, congratulations. Very nice work, very complete, and a lot of work have been done. Also very interesting, and already some of the questions I have been already mentioned. So one of the last things that have been discussed about the creation of the graphs and if this could be automated or not. Yes, I understand. And using LLMs out of the box, it's not easy, but maybe an idea would be to have to train a model as you have already created graphs. You could train a model in order to then work on specific populations that you're interested in. But I guess yes, it's not an easy task. On the other hand, it's quite an overhead for such a system to have to done it manually. And also, I think this causes another question for the whole pipeline and system. It's how you think this can be sustainable and how much effort it needs if you have new patients coming in order to test, the approaches and new literature coming out that can, for the creation of the graph. So what are the steps you think it needs all this approach in order to be able to be more sustainable with, updating with new data and new literature that's coming out so it can be more sustainable?
Okay, thank you for your question. Okay, I didn't think about this before, but because it was really difficult to create the initial graph, I think that with the literature that coming out, and if we go with the two approaches that we discussed before, which DAG we have, it's better to select if you have two options, and I say it's better to select the DAG with the most edges in order to be sure that you cover the biggest part of the knowledge that exists outside. Then with the new literature that coming out and the new outcomes, the only way I can think now it's to enrich the graph with more edges than delete, except, it's very subjective, because if there is it depends also on the what the literature states for. I mean, if it's like a really important outcome that delete all the prior knowledge that we have, which happens too often with science, you cannot ignore it. So I think maybe on the involvement of the expert in the domain is the best way to evaluate what is published, what newly published, and it can be enriched in the graph.
Okay, great, thank you. Another part that I would like to ask is, you have focused, and it's obvious the reason, and I totally understand that you have focused on one adverse effect. But do you think that this approach can be easily generalized in other adverse effects? So do you think that this would work in the same way on other adverse effect, or is it really tailored to the one that you have studied?
So because the pipeline is focused on the structure of the graph, only from this part you cannot have application in another domain if you don't have a new graph. In another
not directly. I understand this. Of course, you'll need a new graph. But do you think that all the approach that you have used, it will be possibly effective? For we cannot know, but what is your intuition?
If we are talking from the pipeline perspective, yes, I think it can be used if if we take only the steps and the tools that are used. Yes, I think can be tested, and it's what I have in mind to be tested also to other cases to see if the framework is reproducible, is generalizable, and can be used and produce valuable results. It must be enriched with more knowledge, more methodologies. For example, as I said in the contributions, there must be new things that must be tested. But I think that after a certain form, it can be tested in other cases. Yes.
Okay, great. And another question I have is that you have doing the different datasets have different sizes. Okay, as far as I have seen. So I'd like to know how this has affected the results you got. So how important it is actually, and how sure we are about the results based on the number of data we have. And the other thing I would like to ask, if there's, I guess that in these cases we have also imbalanced. So the adverse effects are less than the non-adverse effects, and how these have been taken into account. If this plays a role in the whole training of your approaches.
So I didn't use any methodology. Starting from the last part of your question, I didn't use any methodology to balance the data. I use the imbalance as it is. There is in the literature there are studies that are support that the causal machine learning approach. The causal machine learning approach. There is approaches that are more that are better in non-balanced data and approaches that are better in balanced. There is also this in the literature, and based on my results, the different sizes of the data didn't really affect the results because, for example, in the case where we have ibuprofen and ketorolac, in my results, ketorolac in most of the causal machine learning approach, first of all, these two drugs have similar balance in their data, but in my results, ibuprofen is, the results from ibuprofen is aligned with the literature in terms of how strong is the effect to an acute kidney injury outcome. On the other hand, ketorolac, which is also based on the literature in the same family and also has a big effect based on the ranking that we follow as ground truth, didn't produce valid results that are aligned. Ketorolac has mostly negative results, which is a big difference of what we know. So from this small example, we can assume as an initial conclusion that this doesn't affect the size, doesn't affect significantly the results of the causal machine learning models.
Okay, thank you. And I totally agree that you shouldn't balance them because you have to be close to the real work scenario. So I think that was a good choice. Also, in your pipeline, you have done some assumptions. So how important are these assumptions you have? How much do they affect actually the whole?
So causal inference is the biggest example I can tell is regarding randomization, which is something that we don't have in observational data. So it's really important to assume that you cannot have treatments, you cannot have randomized selection of patients for each one of the treatments that you compare. So you can continue in causal inference only if you make this kind of assumption of observational data. This is the importance. You cannot continue if you don't make the assumptions of randomization. And also the last assumption that supports that the observation outcome, you assume that it's the same with the potential outcome. It's one of the most important steps.
Okay, great. And the more general question, after having done all this very systematic review of the literature and having worked all these years on the causal machine learning and have all these results, do you think that causal machine learning is the way to go for pharmacovigilance, or other approaches also seems promising or should be tested? So now we have generative models, graph neural networks, and many other approaches on AI that could work. Based on your experience all these years on this work, do you think it's the way to go?
I don't think that it's one way to go. I think that it's a very important component or decision making. I think that each one, generative models, prediction models, all have their own role in the procedure. But causal machine learning is a key component and can add more value in decision making in the field of pharmacovigilance.
Great. I'm okay for now. Probably gonna go on the second round, so thank you. And congratulations again.
Thank you very much. So now, we can take a few minutes to give the floor to the two directors of the group. Philippe and Alice, you want to say a few words?
Yes, of course. I would not comment on the statistics, I'm too biased. But I really like the discussion. I have a comment to make. There is a reason, I think that there is a reason why these approaches are not really widely applied. The reason is that we should go one step behind, is that people, data scientists, don't really have data. So if you ask Stella or other people doing their PhDs on this kind of topics, how many public databases they will have to work on, the answer would be 1, 2. I don't know if there is another one. Only me comes to my mind. So the first thing that is missed here in this chain is data. Okay, and the approach that is currently being heavily used, being attempted by the European Medicine Association Agency and other organizations is the use of federated data networks. So this is what the EMA tries to do with Darwin infrastructure, and in order to answer pharmacovigilance and other kinds of issues. So I would also like to make another provocative comment. Methodology perhaps is not really that important. I mean, who cares if causal machine learning is used or GANs are used or whatever? Who cares? If the data are if the data are good enough, and if the signal is strong enough, it will be found, even regardless if I would use causal machine learning or GANs or whatever. So the discussion would go back to the original question by Dimitris. How on earth could we reach to the point that we would have good data? And what do we mean by the term good data? And the response would be, again, how doctors include in each step of the process, in the data pre-processing step. Because no data scientist, no computer scientist, no engineer would ever understand why a hospital would decide to lose money by removing, by having operations and have doctors out of the hospital in like 1 day. So, this is something that could only be discussed could only be clarified by doctors, and then it could be, it's crucial before the methodological, before the statistical. That would be my provocative question.
And maybe can a provocative answer on that. And coming back also to your results. So ketorolac, so I put my pharmacologist role is for, without any doubt, one of the best, the most nephrotoxic drugs, and you find no results on that. Do you think why?
So, multiple reasons I can find on that. First of all, it's the data. Maybe the cohort I use is not representative, or the way that I select the patients cannot fit with each one of the cases of the drugs. I have to, because I select patient in the same way for all these drugs, and if I have a more pharmacological perspective when I first design the cohorts, I maybe be careful to focus on other characteristics of the patients that I didn't know at that point. Another thing is that because in ketorolac, especially cases, I really question how I have so bad results. It's because it's one of the good matching. I mean, the populations are in a good match, and the approaches. I mean, I think it's mostly the data. If I can make a quick guess, it can be the data that counts, the representation.
I have another eventual alternative idea on that. And this is more related on the absence of randomization and the measure confounders that you wrote before. I think that no one, any prescriber could prescribe ketorolac in a context of a risk of acute kidney injury. And I'm quite, would say, it's counterintuitive, but I think that this result is great because it is, in some sense, a sort of expression of this kind. We managed the risk before. You don't need causal machine learning models to say that. And probably the first will be your drafter to comment on that, but it is something that we have to take into account. Even with propensity score matching, even if eventually this is risk score matching, once you arrive, and even with the data, patient level data that could be more or less precise, at the end you have the choice to write this prescription or to give this drug that count a lot, I think, and then to be in some sense put it on when we think about causal model. For example, it's less evident for allopurinol. And as you said before, the clinicians didn't think that allopurinol can cause adverse acute kidney injuries. So they can take into account at least less this kind of risk when the side effects quite low.
So basically, what you're saying is that the clinicians selected themselves outside the study.
Hopefully, I think, because we try to teach them also that, so it's oof.
Okay, very complex, right? Thank you. Marie-Christine, would you like to?
Yes, thank you.
Have the last word.
Yeah. So me, first of all, I would like to share the impression that Pantelis just said about the very good discussion, which probably is due to a very good work. And somehow I like the expression to pave the way because as we are on the way, so many doors opens and you say, oh, I didn't think about it, it's very good. So, but you did a lot in 3 years, actually. So you cannot do everything, of course. So you, and okay, so well, what I can say about you is that you really, first of all, thank you very much to have chosen France, because we have this long collaboration with Pantelis, a wonderful collaboration for many years. And we decided to come in France, so very lucky. And finally, you came into a multidisciplinary laboratory, which was exactly the case, the only place to do it. But at some point after the review, you really thought a lot. And finally, it's your own initiative is to go into the causal machine learning. And so we followed you, actually, a lot. So I wanted to really say that we learned a lot with you, and that all what you've done follows a really scientific path. And this, I congratulate you for that, really. So also what I want, something that I would like to say as well is that it was really good to have you in the lab because of you. In the lab we have always a good bunch of PhD students, but I think you had a specific good friends here. You are not the only one, but you are part of one with your sense of humor, with your dignity, your happiness. You didn't seem very stressed the way things you were. So really, I really enjoyed this PhD. I really enjoyed having you as a student. And I'm not sad because you're staying one area. That's good. And I wish you the best for the next, because the next is in 1 year or so.
Thank you.
Thank you.
Thank you, Marie-Christine. So, Stella, I think it's fascinating for me as a clinician to dive into this kind of work. Because indeed, I think it really opens many doors that And I think it's, as you said, to me, it's so important that clinicians could collaborate with, researchers like you to work on this kind of topic. For many different reasons. One reason being probably that it's of course very interesting to do your, fundamental work that you're doing right now, but because you decided to, in the end, keep in mind the interest of the patient, which I thank you for it, of course, on a clinical point of view. I think it's really also very important to keep in mind what the clinicians need. And if you want to really understand that, you really need to work with them, because you really need to, go and see what happens really when you prescribe a drug, the way you think about it, the way you evaluate the risk, and everything you said about, what happens where we're facing a patient feeling like, okay, is it really reasonable to prescribe this or that? What are the risks? And what we really do is take a whole context into consideration. And this leads me maybe to, ask you, I don't know if you will have any answer for this kind of question, but because you focused your work on real world data, and because now in the hospitals, I mean, where we actually treat the patients, we tend to go to a more global evaluation of what we call value-based healthcare, for example. And in the past years, we have more and more included in our the way we evaluate the quality of care, the patient experience, for example. Do you think it would make sense at one point? Of course, if we imagine that you could have these data sets, to be able to include in your work data like experience, related by the patients about that one experience. And I'm mentioning that because the side effect from a drug, of course, it's very simple if you think about the pharmacology and the intrinsic toxicity of the drug. But if you stop there, no, you will never prescribe, for example, cisplatin to anyone, because it's one of the very, very, very toxic drugs. And what's very surprising is that in the past 30 years, I've been a nephrologist, we never stopped prescribing cisplatin because it's very useful in the field of cancer. And despite we've had, dozens of new drugs in, immunology and all the discoveries that we've had recently, patients still receive cisplatin because it's useful. So if we would stop to the intrinsic toxicity of the drug, we would not prescribe it, and we still do it. And the experience of the situation is so multifactorial. This patient will have acute kidney injury with cisplatin, this patient will not. And I think it's very interesting to open up, this reflection about working on very big data sets, opening to artificial intelligence. But I'm just wondering whether, are we moving to a place where a side effect of a drug is going to be an evaluation of the whole context and the whole experience of everybody in the circle, the pharmacologist, the clinician who prescribes, the patient. And are we moving to a place where we will really be able to evaluate the risk at first? Because this is the bottom line of what we expect to be able to do. It's to make a decision aware of the risk for this particular patient. So how do you think this reflection can, it's linked to the work, the beautiful work you've been doing for 3 years, and maybe it's only, but it's maybe far away from the results you're showing us today, because what I understand is that there's a lot to do. But still, do you think one day we will really change the way we prescribe the drugs?
So thank you. It's a very complicated question for me because
I said I didn't expect it.
No. I was
just
trying to answer because it's also very interesting. And so, as you said, also as you last mentioned, my work is need a little to mature a lot in this first initial stage. So in that stage, I didn't think about this direction, but even I think that if we are able to have even more information for the patient based on their experience, based on the questionnaires. It's can only be helpful. And I mean, if you ask me from the data science perspective, more data, it's a really treasure for someone that works in data science, because more data means that you have more complex, you have more information for a case study, and you are questioning how we're going to produce the most valuable evidence. So I think we have a lot of tools, and I think we are not far away from that stage, because it has to do only with our ability to collect this kind of information. If we're able to collect, we're gonna process, we're gonna produce results. Maybe not the best, but we can start working from that direction. But it's really important. The question is how we're gonna get access to this kind of information. This is the most important. If we enter in your everyday clinical practice, you are able to include, and you have, I mean, your work is so time consuming. If we're trying to even automate somehow a collection of this kind of data, it will be the next steps. It's part of the procedure. We will try to find out how we're gonna use them to produce the evidence. But first, we have to specify what kind of data and how we're gonna collect them. This is the most important part for start working on in this direction.
Okay, thank you.
Thank you very much.
So now, should we, now, do you still have any more questions? Because it's now or never.
Well, now
Do our colleagues online, do you have other comments or questions that you would like to add? No? Okay.
So, yeah, so then we are going to leave the jury
Yes.
in a different room.
Oh, okay.
And we'll come back. Maybe.
Maybe.
Okay, but before you start,
Alors.
No, it's bigger. Biggest one. Okay. And there is an echo.
Is it moving?
No news.
It's mute?
Yes.
And maybe they are not mute. I cannot. No, okay. The delay, yes. Yeah, is there still an echo? I touched something, yes it is, an echo from, it's okay? Can you hear so that I can, yes, can you speak, sorry?
Yes, yes, can hear you very well.
It's perfect.
Say.
Okay. So, Stella, the jury has been deliberating about your presentation today and about the work you've been writing down in your thesis. So we want to share with you a few appreciation of your work. First, it's only about congratulating you about what you've been doing, so you can breathe. Everybody, we all agree that you really did a very comprehensive presentation. It's been clear, very easy to understand for all of us, even those who are not specialists in the field. And that's a nice quality for scientific people. Very pedagogical, well structured, and more importantly, we all had the feeling of the qualities you have as a scientist. And what I mean here is that we all share the feeling that you really have skills in the way you know what you know and you know what you don't know, and you are really open to discussion. And in the way you've been presenting your work, it was very clear that your integrity in science is really strong, and it was really important for the jury to notice that, and everybody shared that. So we really think that your qualities really fit with the beautiful career of in science and the field of research, and we would like you to do that, because I think it would be great for the field in which you are really working now. We also want to say that you've been very talented in discussing and answering the questions. And we had the feeling here that we were very keen at learning from everybody, sharing, and taking what's interesting for you to go further after this very important step, and go on with all what has been shared today with everybody. And I think it's really critical when you do science not to be only focused on what you believe in, but being really open to discuss and criticize your own results. So we really want to congratulate also you for that. It's a very nice posture for a researcher. So in the end, my colleagues said it was an exemplary presentation. So you can really take it for you. It's beautiful compliment. Nice slides. Even in the way you've been writing down your results in your thesis, we all of us, we felt that it was beautifully done, very clear, very nicely written, very well organized. So what else? We're very proud of the work that you've been sharing with us today. We want to thank you for that. And now, for the excellence of your work, I want to ask everybody maybe to stand up, because it's a very important moment for you now. Because the jury and all of my colleagues, we all agree to give you the title of Doctor in Science in the field of medical informatics. And then you really need a round of applause here. Like when you go into a concert, you can measure the quality of your work to the length of the applause. So thank you for that. We need you to read something clearly in English. Did you get it?
Yes, I did. I did. Yes.
So we'll listen to you as a doctor.
So, in the presence of my peers, having reached the completion of my doctorate in medical informatics and having thus engaged in my pursuit of knowledge in the demanding exercise of scientific research, cultivating intellectual rigor, ethical reflexivity, and respect for the principles of the scientific integrity, I pledge, insofar as it depends on me, to maintain an upright and honest conduct in my relationship to knowledge, in my methods, and in my results throughout the rest of my professional career, whatever the sector or field of my activity may be.
Thank you very much, Stella, for the excellence of your work. Thank you to our colleagues online. Thank you to the members of the jury, Pontelis and Marie-Christine. I think everybody's been very proud and happy to share this moment with you today. So enjoy now.
It was my honor to finish my PhD with all of you. Thank you very much. The jury and my supervisors, and also my colleagues, my friends and family that come here from Greece. And yeah, I mean, it was a really long and complicated journey, and I couldn't imagine a better end of this journey. Thank you very much, all of you.
Enjoy the new stuff.
Pictures. Anybody want to get pictures with the jury? Yeah, want everyone? You stay online because you'll be on the pier, yeah. Yeah, you love. Right. Do you see everyone? Yes.
Yeah, well, thank you.
Thank you. Okay. Thank you very much.
Congratulations.
Yes, congratulations. Thank you.
Thank you for being here today online. Thank you.
So you missed the follow.
But you can do
Okay, I guess we can.
Yes, I guess so.
Okay, bye. Congratulations again.
Okay, congratulations.
There is a buffet with wine and Greek food. So if you want, you can stay and enjoy. Thank you.
Thank you.
Thank you, bye.
Merci beaucoup, Anastasia.
Thank you also. Congratulations, Stella. Bye.
Thank you very much.